Transformation. Introduction of plasmid DNA and recombinant DNA technology. For the transformation experiment, you will need plasmid DNA, which is Puck18, E. coli, microcentrifuge tubes labeled positive and negative, and cold calcium chloride. The positive tube will contain your Puck18 plasmid. The negative tube will contain water as a control. Calcium chloride will be used to make E. coli cells competent for DNA uptake. Once the plasmid and water have been added to your microcentrifuge tubes, the tube should be kept on ice at your lab station. Transformation of bacteria, although easy to complete, does require several steps. The first step is to make the E. coli cells competent. This is done using the 100 to 1000 micropipette, which dispenses between 100 and 1000 microliters. You will need to set the micropipette to 500 microliters. Add 500 microliters of cold calcium chloride to the tube labeled E. coli. This will introduce the calcium chloride to the E. coli slowly. After adding the 500 microliters of calcium chloride, gently tap the tube to mix. Once you have mixed the E. coli with calcium chloride, you will then remove 500 microliters of the E. coli and place it back into the glass calcium chloride vial. You will need to do this twice since there is 1000 microliters of E. coli solution in the microcentrifuge tube. All of the E. coli will be added to the cold calcium chloride. Calcium chloride will make the E. coli cells competent. Competent cells are cells that are ready for the uptake of DNA or plasmid DNA. In transformation, bacterial cells will transform or take in extraneous DNA found in the environment. In this case, we are providing the cells with plasmid Puck18. The E. coli cells will need to sit on ice for approximately 15 to 20 minutes prior to use. For the next step, you will need the 20 to 200 micropipette. This dispenses, of course, between 20 and 200 microliters. You will set this micropipette to 100 microliters and dispense competent cells into the positive microcentrifuge tube and the negative microcentrifuge tube. The positive tube again contains your plasmid Puck18 and the negative tube contains water as a control. Once the competent cells have been introduced to the plasmid in the positive DNA tube, the uptake of the extraneous DNA will begin. This is the process known as transformation. In this case, the plasmid being taken up is Puck18. Puck18 plasmids contain a gene for ampicillin resistance. This E. coli in its standard form or non-transformed form is susceptible to ampicillin antibiotic. However, the Puck18 plasmid will introduce the gene for ampicillin resistance. This will allow the E. coli cells after transformation to grow in the presence of ampicillin. They will be resistant to ampicillin. Transformation is just one form of vertical gene transfer thought to contribute to the growing problem of antibiotic resistance in bacteria. Some bacteria will be very selective as to the DNA that they take up, and others, such as Staph aureus or Streptococci, are not as selective and will take up DNA from any other species of bacteria. This greatly contributes to the ability of bacteria to transfer resistance factors from one organism to another, or from one species to another. Once the competent cells have been introduced to the positive and negative tubes, gently tap the tubes to mix the E. coli with the plasmid and water, and then allow the tubes to sit on ice for about 10 to 15 minutes. 15 minutes is the preferred time, since during their time on the ice, the actual uptake of DNA is going to occur. You may want to use a timer for this part of the experiment. After the tubes have been on ice for 15 minutes, Place them in a heat block or water bath set to approximately 37 degrees Celsius for 5 minutes. This is known as a heat shock and will allow healing of the pores put into the bacterial cell walls and membranes by the cold calcium chloride. For the next step of the experiment, you will again need the 100 to 1000 micropipette. You will need to set it to 700 microliters. For this part of the experiment, you will now add 700 microliters of sterile nutrient broth to each of the microcentrifuge tubes. This will provide the nutrients necessary for the recovery phase of the experiment. It is during the recovery phase that transcription and translation of the PUC18 plasmid will begin. Remember, the PUC18 plasmid contains a gene for ampicillin resistance. This means that those organisms or cells that appropriately took up the PUC18 plasmid will begin transcribing and translating it as part of its normal genome. This will instill ampicillin resistance in those bacterial cells. It is important at this stage of the experiment to always handle the negative tube or control tube first. This will help prevent contamination of your control with the already transformed bacteria cells found in the positive tube. After the transfer of the nutrient broth has been completed, you should gently tap the tubes to mix the E. coli cells with the newly introduced nutrient broth. After the tubes have been mixed, they will be transferred to a heat block or water bath set to 37 degrees Celsius. They should remain in the heat block or water bath for approximately 20 minutes. It is during this time, again, that the transcription and translation of the PUC18 plasmid will begin. Once the timer has gone off, remove the tubes from the heat block. You are now ready to begin inoculating plates. The media used in this experiment is nutrient agar that has been supplemented with ampicillin antibiotic. You will need 2 plates, one labeled positive DNA. This will be used for the positive microcentrifuge tube, and negative DNA. This will be used for your control, or the tube labeled negative. Again, notice that we are first handling the negative DNA. For this part of the experiment, you will also need the 100 to 1000 microliter micropipette. It should be set to 500 microliters. You will dispense 500 microliters from the negative DNA tube into the nutrient agar ampicillin plate labeled negative DNA. Do not touch the tip to the agar surface. Immediately after dispensing the bacteria onto the nutrient agar plate, you will need to spread it using a spreader bar. The spreader bar is dipped in alcohol, then run through the Bunsen burner flame to catch on fire. Once the low blue flame has burned off of the spreader bar, it is ready for use. Do not heat or try to sterilize the spreader bar by allowing it to glow red in the Bunsen burner. Once the spreader bar has cooled, you are ready to begin spreading the bacteria. Use the spreader bar gently. Do not push down or put pressure onto the agar, and make sure that the bacteria is spread evenly across the plate. When you have finished with the spreader bar, replace it directly back into the alcohol. This same process will be repeated for the positive DNA plate using the positive tube. 500 microliters is a fairly large volume to be inoculated on a single Petri plate. Make sure after inoculation that you allow the plates to sit on your lab bench for about 10 minutes. This will give ample time for the fluid to absorb into the agar. Once inoculation is complete and the plates have been allowed to sit on the bench for 10 to 15 minutes, they should be placed into a 37 degrees Celsius incubator for 24 to 48 hours. We have found in our lab that 48 hours is the best amount of time to use for observable growth. Shown here is a nutrient agar with ampicillin plate with positive or transformed DNA on the left and the control on the right. Remember, the media being used in this experiment is nutrient agar that has been supplemented with ampicillin antibiotic. For those bacteria that were transformed, they now express the ampicillin resistance gene that is found on the plasmid PUC18 they were introduced or transformed with. The cells that were placed in the control tube were not introduced to PUC18 plasmid. Therefore, there was no DNA uptake or ampicillin resistance gene for expression. These cells should not grow on a nutrient agar plate that has been supplemented with ampicillin antibiotic. Let's review the procedures being used for the transformation experiment. The first step is to make the E. coli cells being used competent by adding ice-cold calcium chloride. After the cells have sit with the calcium chloride for about 20 minutes, you will transfer 100 microliters of competent cells into the positive tube and 100 microliters into the negative tube. You will then place these tubes on ice for 10 to 15 minutes. You will then remove them from the ice, place them in a heat block or water bath set to 37 degrees Celsius for 5 minutes. After the 5 minutes, remove them from the heat block, add 700 microliters of nutrient broth. Then place them back onto the heat block for 20 minutes. After the 20 minutes, 500 microliters from each tube will be inoculated onto a nutrient agar plate containing ampicillin and spread with a spreader bar to form a bacterial lawn. The plate should be allowed to sit at the table lid side up for about 10 to 15 minutes and then placed into a 37-degree incubator until your next lab period. Recombinant DNA technology is important not only in clinical settings or in research, but also in industry, agriculture, and pharmaceuticals. Bacteria are being transformed for all sorts of reasons, from making insulin and many different pharmaceutical products used in a clinical setting, to enzymes used in laundry detergent and food products. This field of biotechnology is growing and will be used more and more in industry and in clinical settings. It is an important tool for a biotechnologist and especially a microbiologist to use and understand.